Hi, I'm Dr. Christopher Farage, a junior doctor from Sydney, Australia, who loves to teach. And today we're going through a tutorial on the structure, function, and production of red blood cells. All right, let's get straight into it. So I guess the first thing to do is to have a look, at a red blood cell from the get-go. So this is what we have. Now, I could sit here and, list out all the structural components of a red blood cell and point out to you all of the uh, key functional advantages that the structure presents. But I don't want you to think of it as like a rote learning list of things, right? Yes, there's no mitochondria, biconcave disc, all of that. And we're going to go through it in due time. But I actually want to come back to that because I want you to understand it from first principles. I want it to make sense. If we can understand what a red blood cell does, i.e., its function, the structure, evolutionarily and physiologically should make sense from that because the structure helps to achieve its function. So I guess I've just put this picture just to kind of like give us an idea of what we're looking at. But I'm actually going to come back to this picture and I'm going to come back to and point out the key structural elements. But before we do that, let's talk about function. So generally, red blood cells can be classified under of classified as having three main functions. The first being, of course, the transport of oxygen. So functions of red blood cells, RBCs. So the first one, and arguably the most important, is the transport of oxygen. Now, how does it do that? So red blood cells contain this complex here called hemoglobin. Now, hemoglobin is present even from a fetus, but the structure is slightly different. So as an adult, as you can see here, we have this protein with four main components. We've got an alpha 1, alpha 2, beta 1, beta 2. We call these globin chains. Globin chains. So each hemoglobin, right, which is the main constituent of red blood cells, are made of four globin chains. And the most amount of hemoglobin that we have is called HbA, hemoglobin A. This is, like I've said, two alpha globin chains and two beta globin chains. There are other types, HbA2, there's the fetal type, HbF, there's sickle cell traits and so forth, which are beyond the scope of this particular talk. But essentially, the majority of hemoglobin, HbA, has these four globin chains. And if we look in close into each of these chains, we can see what this diagram is depicting in yellow as heme, a heme portion at the center of the globin chain. Now, if I zoom out a little bit, you can see it on the left. Okay, heme is the functionally important component of hemoglobin. And as we can see, each hemoglobin has four heme parts. And if you look on the left here, right, we call this a porphyrin ring. Porphyrin ring. And at the center of the porphyrin ring, so just to clarify, this is me zoomed into that heme component. Right? This whole thing is here. At the center, we have an ion, Fe2+, which is important for binding oxygen. So when we say hemoglobin is the main carrier of oxygen in our blood, what we actually mean is that within hemoglobin, there's heme, and within heme, there's iron 2+. And iron 2+ is what can bind to oxygen in order to carry it around our body, carry it through our bloodstream and deliver it to our peripheral tissues. So as you can see, overall, if each hemoglobin has four globin chains and each has four heme components, right? And each has one ion, right? We can see that each hemoglobin is capable of carrying multiple oxygen atoms. Now, oxygen binds to hemoglobin via this fancy relationship. There's what's called a hemoglobin oxygen dissociation curve, which is a bit beyond the scope of this talk. But I guess just generally to say, so if this is saturation and this is partial pressure of O2, that it's sigmoidal in shape, that S shape. And there are different, factors that can affect how tightly oxygen will bind to hemoglobin. pH, carbon dioxide temperature, this protein called 2,3-DPG. But essentially, there are multiple factors that allow hemoglobin to bind to oxygen and then also deliver it later on in peripheral tissues. Perhaps we'll go into more detail in another video. Yeah, this is essentially how RBCs or red blood cells carry oxygen around the body. All right, that was our first function. Second function is transport of CO2. Now, it kind of does this in two main ways. So just for the sake of notes, I'm going to write via HB for oxygen. Now, for carbon dioxide, there's kind of two main ways. There's a direct way and an indirect way. So directly, if we talk about the direct mechanism, carbon dioxide can actually react with hemoglobin to form carb amino compounds. Carb amino compounds. So essentially, carbon dioxide can bind, and it is in fairness, a smaller proportion of how carbon dioxide is transported around our body. Majority is via bicarbonate, HCO3 minus. But carbon dioxide can also be transported by carb amino compounds, which is when carbon dioxide can react with hemoglobin. Now, indirectly as well, and indirect, I'm going to hold off on the thought because it's going to actually tie very well into our third function, which is as a pH buffer. And this indirect transport of CO2 should make a bit more sense once I explain this buffer. So pH buffer, pH buffer of extracellular fluid, ECF. All right, sorry, it's written a bit messy, but actually it's a pH buffer. Now I have a diagram which might help to illustrate how this works. So just to orient you, right? So this circle over here, like going around, that is our red blood cell, and around it is our plasma or our blood. Okay. So if you think about it, whenever we undergo metabolic processes in whatever peripheral tissue, our heart, our muscles, our brain, whatever, we produce carbon dioxide as the waste product of energy metabolism. And so what can happen is that carbon dioxide over here can enter into the red blood cell, right? React with water to produce carbonic acid, H2CO3. And this is via an enzyme called carbonic anhydrase, which I will label for you here. Ca, in this case, is meant to be, is carbonic anhydrase. So carbon dioxide reacts with water by carbonic anhydrase to form carbonic acid, which then can dissociate into bicarb and H plus ions. Now that bicarb can essentially be ejected via this transporter in something called like a hamburger shift. But this bicarb can then be used to as a base and therefore buffers our extracellular fluid, buffers our blood, because the more bicarb we have, the more that can react with H plus, and therefore the more basic or alkaline our blood can be. And so this actually ties into these function 2 and function 3 that we were talking about, because I said it acts as a pH buffer, and as you can see, bicarb is being ejected, which can make our base alkaline, but also it's got that indirect transport of carbon dioxide because the carbon dioxide actually entered and broke down into that buffer. So carbon dioxide transport has actually this direct and indirect effect, whereas the pH buffer is a third bonus, I guess, or a third also independent function. I also note, if we go back to the diagram for a second, I want you to note that the H plus over here, um, the H plus that was produced when carbonic acid um, dissociated can actually react with a histidine residue in hemoglobin um, to form more compounds over here. H plus binds the histidine side chains, which also therefore buffers the H plus that's produced. So we have a two kind of bifactorial method for the buffer of hemoglobin. All right. So there are functions. And so essentially, we're trying to allow our oxygen to be carried around our body. We want our carbon dioxide to be carried around our body so we can get rid of it, right? We want, obviously, our blood cells to go to our lungs, oxygen breathe in. We want it to go into our red blood cells via that iron, the interaction with iron in our heme protein or the hemoglobin protein. But then also, we want to go to those tissues. We deliver the oxygen to the tissues by something called like the Haldane effect, which helps to explain that. Again, that'll be probably another video. We deliver our oxygen to our peripheral tissues. Carbon dioxide is going to come in, we can have our buffer and so forth, travels around our blood, and can either be buffered or breathed out at the lungs. All right. Now, so if you think about it, let's go back to our structure. Well, what do we want from our red blood cells, right? Well, there's a couple things we want. One, we want to be able to fit as much hemoglobin as we can. If the primary functions are to, if the primary function is to carry oxygen around our body, and to carry that oxygen, we need hemoglobin, then ideally, the best case scenario is to have as much hemoglobin as we can. That way we can carry as much oxygen as we can. And so a unique structural feature is that the red blood cells have no organelles. In fact, it doesn't even have a nucleus or a mitochondria or a mitochondrion. So no nucleus, no organelles. It literally kicks everything out, and we're going to have a closer look at erythropoiesis, the production of red blood cells soon. And we'll see this happens right at the end step of our RBC maturation. 2, if you have a look at this side shape, see how it kind of dips in? It's not this flat, kind of homogeneous side. It dips in like this. And on the bottom, it does the same thing, right? This is called a biconcave shape. Biconcave. Break this down, the word, so it makes sense. Bi, two, prefix bi meaning two. Concave is, that like concavity, the idea of kind of dipping in. And so biconcave is because it dips in on both sides. And the reason it does that is to maximize surface area. And again, this should make sense. If our whole aim is to allow as much gas to diffuse as we can, to carry as much oxygen as we can as the primary function, we want to have as much area as we can that can be exposed to get that oxygen in. And so if you think about it, rather than having like a flat edge, if it curves in, then you get all this extra area, which allows more gas to diffuse across. And that's a bonus, that's a win. So again, the structure should make sense. And this is why I didn't just list you these things from the beginning, because it should make sense physiologically from its function. Other things, you can say that the membrane is flexible. Why is this of benefit? So flexible membrane. This one is, to be fair, less intuitive, but if we know that our red blood cells are going to be transported all around our body through narrow capillaries, and it's going to have to go through our spleen, we don't want them to rupture. So if you had this like really, fragile, inflexible membrane, our red blood cell would rupture and we wouldn't get anywhere, right? We don't want that. We want our red blood cell to survive as long as it can, within reason, of course. And our typical life cycle is 100 to 120 days. And so we need it to be flexible. That way, when it travels around the body, when it travels through those, tight capillaries, they don't rupture and they're almost resistant in a way to damage. Um, yeah. Now, another thing to note as a structure is the membrane is very thin. Thin membrane. And again, this should hopefully make sense. If you think about it, if you're trying to get oxygen across a thin piece of paper, you're going to get more than if you tried to get oxygen through a brick wall. The thicker the membrane, the more barrier, like the more impedance there is, the stronger the barrier to oxygen and carbon dioxide diffusion. And you obviously don't want that. So it's actually a very thin membrane. And if you look at it, it's like the whole red blood cell is only 2 micrometers in depth. And again, that allows for easy diffusion. All right. So that's the structure, which kind of very much links to the function. Now let's talk about its production. So I have a couple of diagrams here. Let's start big picture, and then we'll zoom in a bit closer in more detail into the production of red blood cells, also known as erythropoiesis. Poiesis, meaning production of, erythro, like erythema, like erythrocyte, erythroderma, erythro meaning red. In all of those words, erythro means red. So, erythropoiesis is the production of red blood cells. All right, so this is a big picture schematic and look at all our blood cells actually. Now, today we're only focusing on red blood cells. So we're only looking at a small proportion of this diagram. We're only looking over here actually, the production of the erythrocyte. But big picture, we have our stem cells, our pluripotent stem cells. And there's kind of two main branches. We have a myeloid lineage and a lymphoid lineage. Our lymphoid lineage produces our T cells and our B cells, and our natural killer cells, whereas our myeloid progenitor line, our myeloid lineage, produces our monocytes, our basophils, neutrophils, eosinophils, our red blood cells, as well as our thrombocytes or our platelets, as well as mast cells as well. So if we now zoom in, if we zoom into that left hand or into my blue circle, right? Let's have a look in a bit more detail. So here we have a diagram that's actually this whole process, to clarify, fits into this blue circle. So this first diagram is overall big picture, but I want to show you more detail of how we get from that progenitor cell, the common myeloid progenitor cell, to our erythrocyte. We're actually zooming in directly into this part here of our diagram. So, erythropoiesis occurs slightly different for a fetus than an adult. So for an adult, the site of erythropoiesis is our bone marrow. And bone marrow in an adult is confined to our central skeleton, which are things like, our skull, our ribs, our vertebra. There is some in the upper part, the proximal part of your femur and into your humerus, but not really in your other long bones, not really in your tibia, your fibula, not in your radius, your ulna, per se, as an adult. Yeah. And that's because of, progressive, fat deposition and so forth as you grow older. But as a fetus, interestingly, erythropoiesis occurs slightly differently. And the kind of the cutoff that they say is 7 months. So when a fetus is less than 7 months, erythropoiesis occurs in the liver and in the spleen, but mostly the liver. Whereas in an adult, to clarify, I stumbled on my words, I apologize. So as a developing fetus, less than 7 months, erythropoiesis occurs in the liver and spleen. when a fetus is greater than 7 months, it transitions to bone marrow. And um, it starts off in kind of like all your bones, and as you get older, as I said prior, it's confined to your central skeleton and to your proximal tibia, proximal humerus as the main sites. Okay. Now, in this bone marrow, there's kind of a complex process, and this is a scary looking diagram, to be honest, but it can kind of be grouped together into the progenitor cell over here, proerythroblast. You can kind of pair these together as your erythroblasts, your reticulocytes, and your red blood cell. So you have that progenitor cell, and it differentiates into proerythroblasts. So now we're specific to our erythrocytes lineage, to our red blood cell lineage. Now, the conversion between this proerythroblast and erythroblasts, so pro being before, is to do with this this agent, this hormone called erythropoietin. Yeah, erythropoietin. Erythropoietin is produced in the kidneys, as you can see here, in our kidneys. If your kidney can detect that it's low in oxygen, right, that it is suffering hypoxia or a deficiency in oxygen, it releases this thing called HIF, hypoxia-inducible factor. Right? So as you can see, if you have hypoxia, it releases HIF in the kidneys, from the interstitial cells of the kidneys, which release erythropoietin. There's a minor release of erythropoietin from the liver as well, but by far the majority is from the kidney. Erythropoietin then travels to the bone marrow, as you can see, and it helps in the conversion of proerythroblasts into an erythroblast. So that's via EPO. Now, oops, zoomed out too far. After we get to these erythroblasts, we have increasing concentrations of hemoglobin at this stage. Okay. Our hemoglobin matures, we get more hemoglobin, concentration rises. As you can see, our cells getting smaller, but therefore our hemoglobin concentration is increasing. Okay. Now, at the end, we kick out our nucleus. We want to make the most amount of space we can for our hemoglobin. So we kick out our nucleus, as you can see, nuclear extrusion. We kick out our nucleus and we produce a reticulocyte. Our reticulocyte is an immature red blood cell. The reticulocyte is then kicked out into the bloodstream, and then after a day, reticulocytes can usually last up to 3 days. But then in the bloodstream, in the periphery, they mature into a red blood cell, which then survives for 100 to 120 days and then gets destroyed by our spleen. The breakdown of red blood cells will be in another video, in bilirubin metabolism. But essentially, that's what happens. So to look back and summarize what we just said, you have hypoxia, you get HIF, hypoxia-inducible factor, which then tells your kidneys a tiny bit to your liver to produce erythropoietin. Erythropoietin travels to the bone marrow to convert a proerythroblast into an erythroblast. That erythroblast then kicks out its nucleus to produce a reticulocyte, which then matures in the bloodstream to form a red blood cell. And there we have it. That's the production of red blood cells. And this comes now to the end of our video. So to summarize, we looked at the structure, function, and production of red blood cells. We've just kind of looked at the production, so I won't recap that once more. But just as a summary of the previous content, we said that red blood cells are these biconcave single cells that have no organelles, no mitochondria, because we know the reticulocytes kicked out that nucleus. Okay, they have no organelles to feed as much hemoglobin as they can. They have this really thin membrane that's flexible to allow oxygen to diffuse across, and also not ensure that the red blood cell doesn't break down in the periphery. And that biconcave shape allows us to have maximum surface area for the gases to diffuse, which is really important because it explains our function. And we said there are three main functions, transport of oxygen being the most important and the first and primary function. And the way that occurs is by hemoglobin. Hemoglobin is a protein which consists of 4 globin chains. The majority of hemoglobin is HbA, which has 2 alpha and 2 beta globin chains. Now, globin chain, at the center of the globin chain is the heme component. Heme has a porphyrin ring and a central iron 2+ atom, which allows binding of oxygen. Each hemoglobin has, as we said, 4 globin chains, and therefore 4 heme substituents, and therefore 4 iron components. This is important for carrying of oxygen, which can bind to the hemoglobin via the oxyhemoglobin dissociation curve, which will be explored in another video and is out of the scope of this talk. Red blood cells are also important for transporting a carbon dioxide. Now, as I said, most of our carbon dioxide is actually independent of this process and travels in the bloodstream by bicarbonate, HCO3-. However, there's also a minor role via binding with hemoglobin to form a carbamino compound or carboxyhemoglobin. Also, there's an indirect effect via the buffering system, which is our third function of red blood cells. And as we saw in this diagram here, that when our carbon dioxide goes in, we produce carbonic acid, which breaks down into bicarb, which gets kicked out over here. And that's important because if we kick out bicarb, we can allow a buffering of blood because we have more alkaline substance, i.e. bicarb in the system. And then we spoke about the production at the end. We said there were 2 main lineages, the myeloid lineage and the lymphoid lineage. And when we zoomed into the myeloid lineage, we saw that we get a proerythroblast, which needs erythropoietin, the hormone produced by the kidney when hypoxia is sensed. And that erythropoietin produces an erythroblast, which then increases its hemoglobin concentration, kicks out its nucleus to form a reticulocyte, which matures in the blood to form a red blood cell, which lasts 100 to 120 days and then is destroyed at the level of the spleen. And there we have it. I hope you learned something. I hope that was helpful, and see you in the next video.